All right, hi everyone. For those of you who don't know me, my name's Brianna. I am a fifth-year graduate student in Roberta Brambilla's lab. And today I'm going to be talking about my project where I'm investigating the role of astroglial TNFR2 signaling in synaptic and cognitive function in multiple sclerosis. If we can get this to work. Okay. We'll see how this goes. Okay, so to start with some brief background on multiple sclerosis, or MS. MS is a chronic neurodegenerative disorder that affects more than 2.8 million people worldwide. It's a T-cell mediated autoimmune demyelinating disorder, and the main hallmarks include acute inflammation, oligodendrocyte death, demyelination, axonal degeneration, and neuronal loss. So MS manifests with distinct clinical phenotypes. A majority of patients are diagnosed with relapsing remitting MS, and this is characterized by periods of increased symptoms and disability, which is then followed by periods where the patient will either recover completely or plateau before another relapse occurs. And about 50% of these patients then go on to develop a secondary progressive MS, and this is characterized by the gradual worsening of symptoms and disability over time. And then the rarest MS phenotype is primary progressive. This is about 15% of cases, and this is characterized by the gradual worsening of disability and symptoms starting from onset of disease. So despite these differences in clinical phenotypes, all of them are characterized by a variety of different sensory motor deficits. And this can include anything from walking difficulty to spasticity, numbness, fatigue, vision problems. And in addition, most patients with MS also experience some sort of cognitive dysfunction. So about 60 to 70% of people living with MS experience a wide range of cognitive impairments, with the most common being memory loss and problems with information processing speed. And this really represents a major burden for people living with MS because it affects their ability to work and their ability to form relationships in both their personal and professional life. But despite the widespread prevalence of cognitive dysfunction in MS and the impact it has on the quality of life, the mechanisms underlying cognitive dysfunction in MS are not well understood. Yeah, I'm not plugged in. I'm on the screen. Yeah. So the mechanisms underlying cognitive dysfunction in MS are not well understood, and there's no pharmacological treatments to reduce cognitive decline or promote cognitive recovery. So we know that cognition is dependent on proper synaptic function, and it used to be thought that neurons were the only participants in this. But we now know that at the synapse, pre- and postsynaptic neurons interact with glia in this bidirectional fashion. And in particular, we're focused on astrocytes because astrocytes play many roles here, such as regulating synaptogenesis, synaptic plasticity and stability, and the efficiency of synaptic transmission. And they can do this via a variety of different mechanisms, including glutamate buffering, modulating glial transmission, and aiding in synaptic formation and elimination. And a key modulator of these functions is tumor necrosis factor, or TNF. So TNF is an immunomodulatory cytokine that regulates things like inflammation, cancer, autoimmunity, and infections. And it exists in two forms, a transmembrane form, which functions via subtle cell contact, and then a soluble form that's generated by the cleavage of transmembrane TNF by tumor necrosis factor converting enzyme. And so TNF binds to two receptors, TNFR1 and TNFR2, and these receptors differ in their ligand affinity, downstream signaling, and cellular expression. So in neurological disease conditions, soluble TNF signaling via TNFR1 is detrimental, and it leads to things like apoptosis and inflammation, whereas transmembrane TNF signaling via TNFR2 is protective, leading to cell survival, anti-inflammation, and remyelination. And so TNF can modulate synaptic function by acting on astrocytes and neurons, and as a result, it can strengthen glutamatergic synaptic activity, alter synaptic scaling, and modulate synaptic plasticity. And it does a lot of these mechanisms by binding to TNFR1 receptors on astrocytes, which really highlights the importance of astroglial TNFR1 signaling in physiological synaptic transmission. So in disease conditions, the level of TNF increases, and its role turns from a regulatory role to either a neurotoxic or neuroprotective role. And so this increase is seen in MS, where in the cerebral spinal fluid and serum, TNF levels are elevated in MS patients compared to unaffected individuals. MS patients also have elevated TNF levels at the site of active lesions. And TNF levels have been shown to correlate with the severity of lesion as well as the disability score. So as the disability score increases, you can see the level of TNF also increases. And it also correlates with disease progression. So as the disease progresses, the level of TNF increases. So there was this key paper by Habas in 2015 that showed while soluble TNF to TNFR1 signaling is necessary for proper synaptic function in physiological conditions, in neuroinflammatory conditions like MS, it becomes overactivated, and this leads to long-term synaptic alterations that then lead to cognitive deficits. So briefly to go over kind of the main findings of this key paper, what they did is they knocked out astroglial TNFR1 in all cell types, and then they re-expressed it so that it was only expressed in astrocytes, and that's this model right here. And then they induced an EAE animal model of MS, and what they saw was in this group where we had astroglial TNFR1 re-expressed, shown in red, there was this increase in excitatory signaling compared to the control groups. And this was accompanied by deficits seen in a fear conditioning test, with these mice performing significantly worse compared to controls. So despite the fact that TNFR2 is also expressed in astrocytes, and it's been implicated in reparative and protective processes in EAE and MS, no one has ever looked at the role of astrocytic TNFR2 in synaptic plasticity, both physiologically or in MS. So that led us to first ask the question of, what role does astroglial TNFR2 play in modulating synaptic and cognitive function in physiological conditions? And to answer this, our lab created a transgenic model where we have the conditional ablation of TNFR2 in astrocytes. So this is an inducible Cre recombinase model where when we inject tamoxifen, it results in the specific ablation of TNFR2 from astrocytes. So this work in physiological conditions was started by a former postdoc in our lab, Placido Iliano, and is something that I've continued as part of my dissertation work. We did all of these experiments in both males and females to see if we also found any sex differences. So to start with, we wanted to look at the expression of key hippocampal synaptic proteins and glutamate receptors that are involved in synaptic transmission in plasticity. So there's a lot of graphs here, and I know it's kind of spazzing out, but to go through them, these top three here are presynaptic snare molecules or proteins, and then we have postsynaptic proteins, and then we looked at glutamate receptor subunits of AMPA receptors and NMDA receptors, and then this last one here is a glutamate transporter, EAAT1 or GLAST. So when looking at the male data, the overall thing that we saw is that in our knockout, so in blue, we see that most of these proteins are upregulated in the knockout compared to the control. Then when we look at the females where we did the same proteins, we see that there is some alterations. For example, gluN2A, which is the NMDA receptor subunit, and this glutamate transporter were also upregulated in the knockout compared to the control. And GluA23 subunit complex was actually downregulated, but overall there were less changes in the female proteins compared to the males.
Many times when we look at the males, the data looks a little bit tight.
Mm-hmm.
When you get the females, it looks a little more spread out in terms of specifically what you're looking at. It looks like you've got some significant sex good. So, do you in particular female mice in terms of estrogen cycle or anything like that?
No, we didn't monitor that at all, or really, yeah, no. But as I go through, you'll see that we did find some sex differences, so that's definitely something to look into in the future. So this did show that when we ablate astroglial TNFR2, we have altered expression of these hippocampal synaptic proteins and glutamate receptors. So seeing that a lot of these proteins that were altered were neuronal proteins, we then wanted to determine if this was accompanied by alterations in neuronal number and morphology. So what we did is we counted the number of new neurons in the CA1, CA3, and dentate gyrus of the hippocampus, and then we performed shoal analysis to look at the morphology of these neurons, as well as dendritic spine morphology analysis to look at the morphology of long, thin, and mushroom spines. So going through the male data, we didn't see any differences in neuron number in the CA1 or CA3. However, in the dentate gyrus, we did see that there was more neurons in our knockout compared to the control. This was interesting because we do know the dentate gyrus is a site for neurogenesis, it could indicate some potential alteration in that process, but future experiments would need to be done to really see that. And then looking here at the Scholl analysis we did, so this is showing the number of process intersections at a increasing distance away from the nucleus. So here we're looking in 30 micron increments away from the nucleus. Overall, we really didn't see differences in morphology. We also looked at number of processes, total process length, and maximum intersection radius. We didn't see any differences here. Then looking at the dendritic spines, we quantified the percentage of long, thin spines, which are more developing or degenerating spines that can't form synaptic connections, and then mature mushroom spines, which have high efficacy for forming synaptic connections. We didn't see any differences here as well. And then looking at the female data, we don't see that same difference in the dentate gyrus. Overall, the knockout and control were very consistent in neuronal number and morphology. So it seems that when we ablate astroglial TNFR2, it doesn't really alter the morphology of the neurons. So then we wanted to see if astroglial TNFR2 ablation alters the behavior of hippocampal glia. So we performed these same experiments now with astrocytes in microglia. So here is the male data. So we counted the number of astrocytes in the same CA1, CA3, and dentate gyrus, and we found that there were significantly more GFAP-positive astrocytes in our knockout compared to our control. When we look at the Scholl analysis, we see that overall, as we go away from the nucleus, there's this trend of more process intersections in our knockout compared to our control, with significantly more at a distance of 8 microns away from the nucleus compared to the controls. We also found that there was overall more processes, and there was a higher maximum intersection radius, which is the radius where there is the maximum number of process intersections. So that was about 8 for the knockout and 6 for the controls. We also found a trend where it looks like overall total process length is higher. So this indicates astrocytes that have more elongated and ramified processes, as you can see in the representative image, which is indicative of hippocampal astrogliosis. When we look at the microglia, we also see that there was more microglia in the knockout compared to the control in all these hippocampal regions. However, we didn't see the same difference in morphology. But overall, this indicates that when we ablate astroglial TNFR2, we have hippocampal gliosis that's occurring. So, yeah.
Look at any ECM changes in these astrocytes?
No, I haven't done that yet, no. Okay, so then we repeated this with the females. And this was interesting because we actually didn't see any differences here. So we see consistent astrocyte and microglia numbers in our knockout compared to our control, and the morphology seems pretty consistent with the Scholl analysis. So this is where we really started to see that we're having sex differences with ablation of astroglial TNFR2, altering these synaptic proteins expression and this hippocampal glia behavior more so in males compared to females. So to further evaluate hippocampal glial reactivity, we did this imaging using translocator protein, which is TSPO, and these radioligands that are used. It's the only validated PET imaging technique to quantify microglia reactivity in vivo, as well as some literature saying that this TSPO expression is also seen in astrocytes, so it could be astrocyte reactivity as well. And so because of this, autoradiography methods have been developed to look at this TSPO binding in tissue sections as a way to measure this glial activation. And so we performed this experiment in collaboration with Dr. Jens Michelsen at the University of Copenhagen. So we sent our tissue sections and they performed this autoradiography method. And so what we see here is for the males, so we looked at total section and then hippocampus, cortex, and thalamus. And we found that in all these regions in the knockout, there was this increased TSPO binding, indicating increased glial reactivity. And you can see specifically in the hippocampus, you can see it's really located there with this strong signal of binding. Then we looked at the females, and we also did not see any significant differences here. So we do see a trend in the hippocampus, but overall no significant differences. And what was interesting is if you can see from the representative autoradiograms, overall, there did seem to be more binding in the females compared to the males, which is just showing us even more sex differences that are occurring. Okay, so then, sorry, one second. We wanted to determine if this inflammatory glia profiles associated with changes in immunomodulatory molecules like cytokines and chemokines. So we sent some protein lysate from the hippocampus to our collaborating lab, Kate Lamberson's lab at the University of Southern Denmark, and they performed this cytokine multiplex array. So here we're looking at key pro and anti-inflammatory cytokines and chemokines. And interestingly, we did not see any differences in the males in any of these cytokines and chemokines. The only difference we see is in females, we have less IL-10 in the knockout compared to the control. And IL-10 is an anti-inflammatory cytokine, so less IL-10 could cause some detrimental effects. But when we kind of think about the female data so far, it doesn't seem to have a big effect on anything we've seen. So this was interesting, though, because it shows that this glial activation that we see occurs without corresponding changes in these inflammatory cytokines and chemokines. So seeing that these male knockout mice had these significant alterations in these hippocampal proteins that are involved in synaptic plasticity, and they also have this altered behavior of hippocampal glia, we wanted to see if astroglial TNFR2 ablation affects hippocampal synaptic plasticity, specifically long-term potentiation. And since overall we observed that the changes were more pronounced in male mice, we wanted to focus this experiment on males. So what we see is that compared to controls, we see that the knockout mice had this suppression of LTP. The ESP slopes dropped rapidly to pre-stimulation levels after tetanization. And then when we look at the input-output curve, we see that the response is shifted downward in our knockout, which suggests depression of basal transmission. And then here is the paired pulse facilitation ratio, which also shows this declining trend, which suggests that there's potential alterations in short-term synaptic plasticity as well. So this did show that ablation of astroglial TNFR2 in the male mice in these physiological conditions does impair LTP. So then we wanted to look at cognitive function because cognitive function relies on proper synaptic activity. So we assessed this through a series of behavioral tests. We started out with the Morris-Water maze test. This is a test that investigates spatial learning and memory. And in this acquisition phase, a mouse is placed in a pool and it uses spatial cues to find a hidden platform. And the amount of time taken to find that hidden platform is quantified as spatial learning. Then during the probe trial, that platform is removed, and the amount of time taken to find the platform area where the platform used to be is quantified as spatial memory. And what we found is for both males and females, our knockout group took a significantly longer amount of time to find this hidden platform area compared to the controls, indicating that ablation of astroglial TNFR2 results in impaired spatial memory. We also performed the novel object recognition test. So this is a test that looks at long-term memory, and it's based on the tendency of a mouse wanting to investigate a novel rather than a familiar object. And what we found is male mice lacking astroglial TNFR2 had a significantly more difficult time discriminating that novel from the familiar object, showing that when we ablate astroglial TNFR2, we also have impairments in long-term memory in male mice. Now, we didn't see that same difference in females. And then lastly, we performed the fear conditioning test. So this is looking at contextual-based associative learning. And here we did not see any differences for both our male and female mice, showing that when we ablate astroglial TNFR2, we don't have impairment of contextual associative memory. But overall, in both our male and female mice with knockout of astroglial TNFR2, we do see cognitive deficits. It just seems that the males have this stronger cognitive impairment phenotype. Okay. So lastly, in physiological conditions, we wanted to investigate the role of TNFR2 in regulating intracellular calcium dynamics in astrocytes, because calcium is a key mediator of astrocyte function and synaptic modulation. So we know that elevations in transient calcium levels leads to release of glial transmitters like glutamate and GABA and ATP and D-serine, and then these glial transmitters can act on neurons, and this is a way that astrocytes regulate synaptic transmission and plasticity. So this experiment was done in collaboration with Dr. Laura Bianchi's lab here at UM. And so just to explain what we did, so we cultured primary astrocytes from wild type, which is in gray, and TNFR2 knockout in red astrocytes. And what this graph is showing here is the percentage of fluorophore fluorescence above a baseline fluorescence. And this fluorophore is a calcium indicator that exhibits an increase in fluorescence when it binds to calcium. So these shaded purple areas represent either our vehicle perfusion as in A or our ATP perfusion as in C and E. And then these graphs here on the right are showing the peak fluorophore fluorescence above baseline, and each of these points represents a single recorded cell. So what we found is that in the top row here, in basal conditions with no ATP, we see that our TNFR2 knockout cells have this abnormal, spontaneous, elevated calcium transients. And then when we stimulated these cells with ATP, we saw that these knockout astrocytes had these abnormal peaks that are evoked by this ATP. And so, since we know that elevated intracellular calcium levels is a signal that leads to glial transmitter release, it could suggest that astroglial, or that ablation of TNFR2 may result in possibly altered glial transmitter release that could impact neuronal function in a negative way. So overall, our key findings in physiological conditions, we found that ablation of astroglial TNFR2 results in altered expression of key hippocampal synaptic proteins, glutamate receptors, and transporters in both male and female mice, with there being more alterations in males. We found that there was hippocampal gliosis, an increase in neurons in the dentate gyrus, and suppression of LTP in male mice. We also found that there was cognitive deficits in both male and female mice. And we found that TNFR2 regulates astrocyte calcium transients. So overall, these findings indicate that astroglial TNFR2 is necessary for proper synaptic and cognitive function and regulates this hippocampal synaptic plasticity in this sex-dependent manner. So this data, combined with other data from our lab that shows that TNFR2 is protective in MS and EAE, led to our next question of what role does astroglial TNFR2 play in modulating synaptic and cognitive function in MS and EAE? And our hypothesis is that astroglial TNFR2 plays a protective role in containing or counteracting synaptic and cognitive dysfunctions associated with EAE. So to briefly go over our EAE model, EAE is experimental autoimmune encephalomyelitis. It's the most commonly used animal model for MS. And what we do is we start out on day 0 with a pertussis toxin injection, and this begins to break down the blood-brain barrier. Then on day 1, we do this MOG injection. MOG is myelin oligodendrocyte glycoprotein, and this specific peptide sequence from MOG exposes peripheral immune cells to the protein that's expressed in myelin. So then on day 2, when we do another pertussis toxin injection, all those immune cells that have been primed against myelin will infiltrate and attack, and this mimics what happens in MS. So this results in a locomotor phenotype that we score on a scale from 0 to 6, 0 being completely healthy, 6 being death due to the EAE, and then we have levels of paralysis in between. So essentially, paralysis starts from the tail and works its way up the mouse's body. We have different phases of this model, so we consider from onset of disease to about day 25 to be the acute phase, where scores are mainly driven by the infiltration of peripheral immune cells, which is T-cell mediated. And then from day 25 to the end of the study, we consider the chronic phase, where the scores are mainly driven by neuronal damage and demyelination. So in these EAE experiments that I've performed so far, I have only started with female mice. I chose to start with female mice for these EAE studies because the lack of or less differences in physiological conditions makes it easier to investigate TNFR2 dependent changes that are dependent on the actual MS pathology. And also, I started with females as EAE is usually used with females in the literature. So I'm going to go through this female data in EAE conditions. So again, I started out by just quantifying the hippocampal synaptic proteins and glutamate receptors that we looked at. So started out by looking at these presynaptic snare proteins, so SNAP25. So in addition to this naive time point, now we have pre-symptomatic EAE, which is 10 dpi, and this is before the mice have locomotor symptoms. And then we have acute disease at 22 dpi and chronic at 45. So what we found for SNAP25 is that at this pre-symptomatic EAE time point, we had this increase in SNAP25 compared to our control. When we looked at syntaxin 1, we also see it's upregulated in the knockout compared to the control at this pre-symptomatic EAE time point. And then we looked at synaptotagmin, which is really important here. So these snare proteins are a part of the process of vesicle fusion, which leads to exocytosis of neurotransmitters. And synaptotagmin acts as a calcium sensor, only promoting that vesicle fusion when there's high levels of calcium in the cell, so making it a really important protein. And we also see that it's upregulated in the knockout compared to the control at pre-symptomatic EAE. So overall, we see that we're seeing these changes at this pre-symptomatic time point before the mice even have locomotor symptoms. So then I looked at postsynaptic proteins, PSC93 and PSC95. We did see a slight decrease in PSC93 at acute disease in our knockout compared to our control, but overall really no differences in these postsynaptic proteins. Then we looked at the glutamate receptor subunits. So first, we are looking at the AMPA receptor subunits. So AMPA receptors are critical for mediating glutamatergic synaptic transmission, and they're made up of 4 subunits, GLA1 through 4. And depending on the composition of these subunits, it can affect the function of these receptors. So first, we looked at GLA1. GLA1 containing AMPA receptors have been shown to play an important role in synaptic plasticity, like long-term potentiation. And what we see here is that, again, at pre-symptomatic EAE, in our knockout, it's upregulated compared to our control. And then it goes down here at acute disease. Then we looked at GLUA2-3. This is a subunit complex. And specifically, GLUA2 is important because it controls the permeability to calcium. So AMPA receptors that contain GLUA2 are impermeable to calcium, and receptors that lack GLUA2 are permeable to calcium. And so this GLUA2 subunit does play an important role in calcium homeostasis. And so, as I showed before, in the physiological conditions, it is downregulated in our knockout. But then we see at pre-symptomatic EAE, it's upregulated, and this persists throughout acute disease. And then lastly, we looked at GLA4. This was also upregulated already in naive conditions, which persisted throughout pre-symptomatic EAE. So looking at these two, we really see that a lot of these alterations are all happening at this pre-symptomatic EAE time point. Then we looked at the NMDA receptor subunits. So when NMDA receptors are activated, calcium can enter the postsynaptic cell, and that is required for many cellular processes and is essential for synaptic plasticity. And NMDA receptors are made up of at least 2 different subunits, gluN1 and gluN2, and gluN2A and gluN2B are the predominant subunits in the hippocampus. So first we looked at gluN1, and we didn't see any changes at any of our time points here. Then we looked at gluN2A, which was upregulated in naive conditions, and also at pre-symptomatic EAE. And then gluN2B, which was also upregulated at pre-symptomatic EAE, as well as acute disease. So NMDA receptors that are located at synaptic sites are enriched in gluN2A, whereas NMDA receptors at extrasynaptic sites are enriched in gluN2B. And this subunit is very important because it's been shown to have altered signaling when hyperactivated that could lead to synaptic damage. So building on that, we then wanted to see or investigate dendritic spine morphology because dendritic spines are the primary recipients of the excitatory input in the brain, and the morphology will provide us important information on the functional state of ongoing transmission. So we analyzed dendrites in the stratum radiatum of the CA1. And first we looked at spine density. So here we're looking at naive, pre-symptomatic, and chronic EAE. We didn't see any differences in spine density. And then we quantified the percentage of mushroom spines, which are mature that can form synaptic connections, and thin, long thin spines, which represent degenerating spines. And what we see here is in our pre-symptomatic EAE condition, we saw significantly less mushroom spines and significantly more thin, long, thin spines, indicating that we do have synaptic damage occurring at this pre-symptomatic EAE time point. Okay, so seeing this, and as I previously said, the hyperactivation of this gluN2B subunit has been shown to be associated with causing this synaptic damage. We wanted to see if we inhibit this gluN2B if we could rescue this altered dendritic spine morphology at this pre-symptomatic EAE time point. So we performed this experiment. This was done by Taylor Pohl, a new graduate student in our lab. And here is our experimental timeline. So the mice were injected with tamoxifen as usual, EAE was induced, and then mice were injected via IP with 5 milligram per kilogram of this romaliate drug, which inhibits glu-N2B. And this was dosed at 4, 6, and 8 DPI. Then our tissue was collected at 10 DPI, which was our pre-symptomatic EAE time point. We processed half that brain to do Western blots, and then half was processed for the Golgi-Cox staining to do the dendritic spine analysis. So here we just looked at glu-N2B expression. So to go over these groups, our white is our control, our dark pink is our knockout, and then our gray is our control with the romaliate, and our pink is the knockout with the romaliate. And so what we see is we saw in our knockout that it was upregulated compared to our control. But when we add this drug, it brought it back down to kind of baseline conditions, more comparable with the control. Then we looked at spine density. We didn't see any differences here, but we didn't see any differences to begin with, so we weren't expecting that. And then when we quantified the thin, long, thin spines and the mushroom spines, we saw that while in the knockout there was more thin and less mushroom, with this inhibition of glu-N2B, we had a rescue of this morphology with the percentages of these spines being more comparable to control conditions. So this did indicate a protective role of glu-N2B inhibition at this pre-symptomatic EAE time point. What was interesting is we also quantified the other receptor subunits, and so all of these were upregulated in our knockout compared to our control. And with the administration of this romaliate drug, all of them were brought back down to baseline or even lower levels, which was really interesting to see. And then another thing we did is we quantified the number of GFAP-positive astrocytes now in our EAE condition, so at pre-symptomatic, acute, and chronic EAE. And once again, we found that at this pre-symptomatic EAE point, we have alterations with there being more GFAP-positive astrocytes in the knockout compared to the control at this time point, indicative of hippocampal gliosis. So this is just a little cartoon that kind of goes over what we see at this pre-symptomatic EAE time point and potential hypotheses of what could be occurring here. So on the left here, we have our normal mouse with normal synaptic transmission and plasticity. In our knockout, when we knock out TNFR2 from astrocytes, we see that there's this gliosis, so these astrocytes become reactive. They could increase in their intracellular calcium levels, causing altered glial transmitter release of glutamate and D-serine, as well as other glial transmitters that could bind to the presynaptic cell and trigger a cascade of excitotoxicity. We see upregulation of all of these proteins involved in synaptic transmission, which could be detrimental. And we did see that this led to altered dendritic spine morphology, and then we saw that this spine damage was rescued when we inhibited the glu-N2B subunit. So lastly, we wanted to look at the behavior. So we performed these same learning and memory tests now at pre-symptomatic EAE and chronic EAE. And so here we're looking at the novel object recognition test. Again, this looks at long-term memory. So here we found at chronic disease, the mice lacking astroglial TNFR2 had a significantly more difficult time discriminating the novel from the familiar object, indicating that we do have a worsening of long-term memory at this chronic time point. Now, it was interesting that we didn't see any changes at this pre-symptomatic EAE time point where we see all these alterations. One thought is that maybe these changes are occurring, the cognitive deficits could occur after we see these synaptic alterations. I did try to do some behavior following pre-symptomatic EAE, but it is very hard to test cognitive function with mice that have locomotor deficits, so I wasn't really able to see that. But we do see at chronic disease, we see these deficits. Then we perform the fear conditioning test. So here we're looking at both context and cue-based fear conditioning. So just to briefly go over this, so in this training phase, a mouse is placed into the apparatus and a cue is played, and then a foot shock follows that. And then during the testing phase, the mouse is placed back into the apparatus. If it's learned to associate the apparatus with the foot shock, it will exhibit increased freezing behavior, and that's contextual-based associative learning, which has been shown to be more hippocampal dependent. So here, again, we see at chronic EAE, these knockout mice performed worse. They froze less compared to controls. And then cue-based associative learning. So in the second part of this testing phase, we changed the apparatus to have new walls, a new grid, we introduce a new smell, and then that cue is played, and the amount of freezing after that cue is recorded as cue-based associative learning, which has been shown to be more amygdala dependent. And once again, here at chronic disease, we see that these mice performed worse compared to controls, indicating that astroglial TNFR2 ablation also leads to a worsening of associative learning and memory at chronic EAE. So in addition to these cognitive tests, I've also taken the daily clinical score from these animals. So this is showing our EAE curves. So here on the left is the EAE score from 0 to day 40 of the study. The right is the same data, but we're starting from day of onset because each of these mice has a different day of onset. So this is easier to see disease progression. And overall, we see that the knockout group has a worse EAE clinical outcome compared to the control. They have a higher cumulative disease index, which is a summation of all the scores. They also have a higher peak disease score. But it is important to note that the EAE score is a measure of locomotor function that mainly results from immune cells infiltrating into the spinal cord. And this is likely due to different mechanisms than what's happening in the hippocampus with the synaptic and cognitive alterations. So this is not the main topic of my project, but is something we are further studying. For example, one of the things we are investigating now is seeing if our knockout mice maybe have more impaired blood-brain barrier function, leading to more immune cell infiltration that causes this worsening EAE clinical outcome. So lastly, we asked if ablation of astroglial TNFR2 leads to a worsening of cognitive function in EAE. Does upregulating astroglial TNFR2 signaling rescue this phenotype and improve cognition? And so to answer this, our lab created a transgenic model for astrocyte-specific overexpression of TNFR2. This is a TET-off doxycycline-dependent model that results in TNFR2 overexpression in astrocytes. So here we performed the same cognitive tests. Looking at the novel object recognition tests, we didn't see any significant differences, but at chronic disease, we see a trend with these overexpressor mice performing better compared to controls and performing closer to more naive conditions. Then we did the contextual and cue-based associative learning with the fear conditioning tests. And once again, we see at this chronic time point that these overexpressor mice here with the context perform significantly better compared to controls. And we see that same trend in cue as well. So it does show us that overexpression of astroglial TNFR2 does lead to improved associative learning at chronic EAE, and we have somewhat of a rescue of what we saw with our knockout. Once again, I took the daily clinical score for these, and we don't see any differences here, which was expected. So overall, our key findings in EAE conditions, when we ablate astroglial TNFR2 in EAE, we see altered expression of hippocampal synaptic proteins and glutamate receptors. We see this altered dendritic spine morphology at this pre-symptomatic EAE time point, and pharmacological inhibition of gluN2B with Rho malleate rescued this spine morphology. We see hippocampal astrogliosis at this pre-symptomatic time point, and we see cognitive deficits at chronic EAE, and these were rescued with astroglial TNFR2 overexpression. So altogether, this data shows that astroglial TNFR2 signaling is essential for proper synaptic and cognitive function following EAE. Okay, and with that, I'd just like to thank my lab, all of the students who have helped with this project. It's been a really collaborative effort, and all of our collaborating labs as well, my committee, and all the cores here at UM. And I'm happy to take any questions. Thank you.
So, going back to the baseline differences that you saw in young mice, so it's really interesting that upregulation of classical glutamate receptors and you had changes in synaptic and spine numbers, but yet you had a decrease in LTP and a decrease in learning number 2. And I do know what's going on there.
Yeah, that's something I also noticed and is kind of the confusing part of this. I'm not exactly sure. it could, yeah, I'm not exactly sure. I don't know. I kind of want to redo that dendritic spine analysis to see if that actually is true, because I did expect to see impaired dendritic spine morphology, but I'm not sure. Maybe there's some other mechanism that's not exactly altering those dendritic spines, but is still causing suppression of LTP. But that's something I still need to think about. Yeah.
Thank you.
Okay. Thank you.